genes, their expression regulates. Yes, they don't code for proteins, that's true. Do they look like defunct copies of other genes? Yeah, they do. But you have two papers back-to-back in Nature, and what they demonstrated is that, well, those so-called non-functional RNAs, the transcripts, are regulating other genes. And I'll be happy to provide you with the PDFs and with the references for those papers.
Okay. I have a comprehensive list on page 407 of my book of the functions of non-coding DNA.
I just want to say that we had some wonderful questions up here, and I'm very sorry I can't get to all of them. But I have chosen one for each of the participants tonight, and we'll start with one from Michael Shermer. Can Michael Shermer provide just one example of random mutations erecting whole new genetic information that leads to a new vertical species?
Well, sure. First of all, natural selection is not random. The best definition, I think the T-shirt definition of evolution is from Dawkins, it's random mutation, non-random cumulative selection. It's the cumulative selection part that drives evolutionary change. One example amongst many, the origins of complex eukaryotic cells. This is the research of Lynn Margulis and her lab, in which they've demonstrated that things like the kinds of cells that you and I are made out of, eukaryotic cells, in fact have within them organelles that were once prokaryotic cells, simpler, self-sustaining organisms that then combined into this sort of non-zero cooperative process that gained a selective advantage and became more complex. Dr. Margulis has shown over and over again that these simpler organisms acquire entire new genomes. Just like that, they double their information content and double it again and double it again. And you can watch it right there in the lab. And you can see it in our genetic fossils. You can see it in the mitochondria of your own eukaryotic cells has within it DNA. Well, we already have a complement of DNA in the nucleus of our eukaryotic cells. Why would there be another set of DNA in our mitochondria? The answer is because those mitochondria used to be simpler prokaryotic cells. So that's just one example among many. Thank you.
Okay. To the ID side. If mutation and selection is not sufficient and there is no external influence, then what mechanism active is responsible for new evolutionary forms?
The question of mechanism is a subset of the question of cause. And where we invoke an intelligent cause, we invoke that in place of a materialistic mechanism. A mechanism is just a material cause. So what ID does is it infers the activity of a designing intelligence from the presence of effects which are known to arise from one and only one type of cause, and that is intelligence. So ID does not provide a mechanism, but it does provide a cause, and that cause is intelligence.
Thank you. Dr. Prothero. Prothero, sorry. You stated that labs are close to creating life from non-life. Without the information, blueprints, the inherent genetic information already present, explain how it's possible to create life devoid of pre-existing information?
There are actually several labs on this, and you can Google this and read it up on your own if you want. A recent paper in New Scientist talked about one of these labs in Britain. The Venter lab, the same guy who privately sequenced the genome of the human, is also doing something similar to this. And basically they're doing a very reduced system where they're doing the minimal amount of genetic code needed to make something reproducing, copying itself faithfully. And they've got quite a sophisticated setup which is working at the moment quite well. They haven't gotten to the point where it reproduces completely in a full living sense, but they're already at the point where they have a lot of the pieces of the puzzle fit together. So it's happening. It's just we don't have the time in the diagram to show you all the biochemistry for it.
I address that in my book. That's called the intelligent engineering of new genetic information.
The intelligent design side, is the fossil hominid evidence, along with human-chimpanzee genetic commonality, sufficient enough evidence for you to accept a neo-Darwinian explanation of human origins?
I'll respond to that. What I would say is this. I and I'll speak for myself, I accept the hominid sequence. I do not accept a neo-Darwinian explanation for it, mainly because, given, again, the breeding population size and given the repeated bottlenecks that the population, the lineage supposedly went through, and again, I accept that, what you're left with as a causal mechanism is not natural selection but genetic drift. This has been repeatedly shown. Ira Walker has done work on this. Michael Lynch has written extensively about this. So when you look at the sequence, pick an ancestor, Australopithecines or Arty, what have you, and you want to walk through until you get to Homo sapiens. If you accept that sequence, what you're left with as a causal mechanism is essentially what's called a neutral or near neutral process, or what's essentially a random process. So I think and again, this goes to the testable hypothesis, the opposition seems to think that if you present a theory and you have an examination of that theory and you say, well, this aspect of the theory is found wanting, then you've automatically lapsed into some supernaturalism. Well, that's not the argument I'm making at all. I'm saying that if you accept the sequence, the neo-Darwinian mechanism, to my lights, does not appear to be sufficient because you have to invoke something like genetic drift.
Rick, isn't it also the case that the non-coding regions of the genome in humans and chimps are species-specific? So that old argument about 99% of human and chimp DNA is identical is that the 99% of the 2 or 3% that's coding, isn't it?
Well, first of all, roughly about 1.5% of the mammalian genome codes for proteins. That aside, a lot of the alignments that are done, what many people do not take into account is that aside from the fact that the chimpanzee genome, the sequence that is, has not been completed, some regions haven't been sequenced, is that in order to get the alignments that you see, to get the so-called 98% similarity, you have to rearrange the sequences. You have to count for insertions and deletions and rearrangements and things like this in order to get it to add up. And then you also have to factor in that you have a number of species-specific sequences, like retroviral type sequences, and like so-called jumping genes, which reduces that further. And the fact of the matter, and there's a paper that came out in Science, it's called The Myth of 1%, is that we really don't know. And I haven't seen any paper that has come up with a definitive figure. We don't know the actual percentage. That is the actual percentage of similarity between the humans and the chimpanzee. Anyway, and I can find the reference for you. But it's an insightful paper. Some gymnastics has to be done at the sequence level to get them to map on. Now, of course, Dr. Prothero, Dr. Shermer will respond and say, well, yes, of course, and the reason that you have to perform these alignments is because, well, of course, a lot of evolution has occurred. Well, perhaps. But nevertheless, you cannot say we've got such close similarity by taking a few sequences, lining them up, and then adjusting them to make it look like you've got a lot of similarity. The fact is our chromosome complement to some extent is different, but also if you look at the raw sequence, do the raw sequence comparisons, you will find that there are some significant differences between the two species in terms of the genetic complement.
Gentlemen, I have to ask you not to ask each other questions. That privilege actually belongs to the audience right now. You got away with it this time, but you won't again. To Dr. Shermer, do you believe that reason is an emergent property of molecules, and if so, why should we trust anything you say?
Well, first of all, you shouldn't. The first principle of skepticism is be skeptical of the skeptics. So, obviously, but I think there's something deeper, actually, in the question on to what extent should we trust any kind of inductive process practiced by science or reason or logic or something like that. Well, as opposed to what? As I started off saying, there's only two explanations. There's the scientific one based on reason and logic and evidence, and then there's everything else. And the one that works the best, just pragmatically, is science. You want to get a spacecraft to Mars, you use astronomy, not astrology. Just because it works is the best answer to that question.